Earth, and we understand the language of life. 1 second, students, okay? Some protocols. So we understand the language of Earth and we understand the language of life. Is it a good thing? Absolutely best. It is the best experience you can ever have in your life. Biology doesn't mean that we just have to be so descriptive about everything. Oh, tell me this, section of a dicot stem, that dicot root. Okay, draw a neat diagram of this, draw a neat diagram of that. What is a cell? Okay, what happens during this phase of the mitosis or meiosis? 101 questions can be posed to you. And even though it's quite hectic to be theoretical about everything that we speak about, but in the heart, we understand something beyond. So, humans have two gifts. One, to understand the natural world away from this planet Earth in the manner of chemistry, physics, and mathematics. Through mathematics, we try to define so many things that we cannot see, and we try to bring reality to that. With physics, we try to see the possibilities, we try to prove the possibilities, and we try to bring it into the natural world. With chemistry, we're trying to understand the very fabric of how this entire construct of all the living animate and inanimate objects are around us. Correct, right? But why biology? What's so special about biology? We are gifted this wonderful life. And in this wonderful journey that we spend our time, we can have anything we want. Anything. We can have the marks that we desire, okay, or we can buy a car, we can build a home, we can become a leader, we can become a politician, we can become a teacher, a scientist. Irrespective of whatever you become in the life, humans have something more special compared to any other organism on this planet. Agree with me? Why not we act and define ourselves as rest of the animals, like a lion or a dog or a cat or any other animal? Why humans have a special place? What makes us completely different from others? And the answer for that question comes with consciousness. We have a beautiful consciousness to understand what is right and what is wrong. With this common understanding, we go to a very special dimension of understanding what is life. Even though biology defines that it is a study of life, yet we still have to travel a very, very, very long journey to understand life in so many different perspectives. If I ask you somebody what is life, somebody is going to give me a philosophical answer. Life is nothing but a journey from death to life to death, and you don't gain anything, you don't lose anything, you just live through these moments. Correct, right? Whereas if I ask a biologist what is life, a biologist describes it in a very complex manner. It is the selfishness of around 30 trillion cells that is in your body to make you live every single day, to make you perform the activities that you do every single moment, that to make you feel special every single second. You might hate yourself, you might not like yourself, or dislike some of the things that are within you, but yet your cells love you so much that they always give you energy, they'll always give you the rest, they construct your proteins, they destroy the bad things, they clean up the things that you put in your body, right? And also give you this consciousness to understand your surroundings, make friends, be happy, be sad, you name it. There are like thousands of biological functions, okay, that integrate together to make some things possible. Not everything, some things possible. On a whole, we experience something called life. So as a biologist, what else can we ask more than to go deeper into understanding life itself? Philosophy will come to us eventually, but the truth is what we have to search for. Science defines truth. If we ask a definition, what is science? It is nothing but truth. So we go in search of truth. As biologists, we go in search of truth of life. So that is why you guys are special. All right. So Anyway, we have lost a lot of classes, and then we have not got a formal introduction from the living world to all the way till here. But anyways, let's make sure that we have a beautiful introduction with each other, and then we start with this amazing topic called as biomolecules. Okay, so starting with biomolecules, why do we have to study chemistry? Why do we have to study the chemistry of life? Even though it's very easy for you to just understand what are we talking about. We are talking about bio, life, molecules, the life molecules, right? The molecules which make up our life is called as biomolecules in simple terms. Why do we have to study it? Don't we know that how the cell functions? Don't we know how the cells integrate to become tissues? Don't we know the function of our brain and our kidneys, our heart, liver, okay, etc.? We know their functions, yet something is missing. What is missing? What makes up all these things? What is a cell made up of? It's made up of organelles. What are organelles made up of? Organelles are made up of integration of so many different things. What are those so many different things? These so many different things are nothing but biomolecules. If you take a cell and if you examine a plasma membrane, you are going to look up, look into two different types of things will be visible to you. One, you come across proteins. One, you come across lipids. When you go to the center of your cell and you see this darkly stained body in the center of your cell, you come across this special body and you call it the nucleus of the cell, the control unit of the cell. Wow, it's control unit of the cell. What does it do? Look at yourself. So many different characteristics that you have. Wonderful characteristics. Like you, nobody is there in this 8 billion people. You are very unique. Who gave you this uniqueness? How did you get this uniqueness? The chemistry. Where is the chemistry within the nucleus? What kind of chemistry are we talking about? They are called as nucleic acids. What are nucleic acids? In simple terms, maybe I would have heard about the words called as DNA or RNA, deoxyribonucleic acid or ribonucleic acid. All right, so we talked about lipids, we talked about proteins, we talked about the presence of nucleic acids. Some people will tell, I have a cholesterol problem. How did you get cholesterol problem? How is your hair getting a wax? So to answer all these questions, as we go deeper and deeper into the organs to the tissues, tissues to the cells, cells to cell organelles to what they're composed of, we end up with simple chemistry. And these chemical molecules that make up your entire entities of all the cells is nothing but biomolecules. All right. So you can hear and see well, right? You can hear me well and see the board well, correct? Agree with me? Yes, no, students, can you hear me? You can, oh yeah, perfect. You're able to see and hear. Okay, so let's get on. So you gotta let me know if you're okay with the language. Okay, if you're able to understand me. As we move on, we will get it even more better. All right, so let's get on with this. To start with the biomolecules chapter, again, what are biomolecules? They are nothing but the chemicals or the compounds that are found in the living organisms. Simple as that. That is what I defined you just few seconds back. And what are they made up of? What are biomolecules made up of? Biomolecules are made up of either organic or inorganic compounds. So you might have heard in chemistry what are organic compounds. Anything that is made up of carbon and its compounds are called as organic compounds. What are inorganic compounds? For such as ammonia, such as nitrates, such as sulfates, such as any of the ions like magnesium, calcium, iron. So they're all inorganic compounds. Organic compounds can be your glucose or the carbohydrates or the nucleic acids or the lipids, right? All of them are carbon containing compounds. So they are called as organic compounds. And the other type of compounds we see is inorganic compounds. So let's take a deep dive. So what we have to do is perform a simple experiment. So to perform this simple experiment, we need some living tissue. So to find this living tissue, what we are going to do is we are going to fetch some chicken liver, or we are going to find some plant leaf. So we are going to take this chicken liver and the plant leaf. So we are going to take them in mortar and pestle. So you know what is a mortar and pestle, right? Something like uh, it looks like this, which is made up of Chinese bowl material, and then a pestle looks like this, correct, right? So mortar and pestle. So what we do is we add our living tissue, either a piece of liver, or we are going to take the leaf, and to this living tissue we are going to add a chemical called as trichloroacetic acid. What are we going to add? Trichloro trichloroacetic acetic acid. So why do we add trichloroacetic acid? Why don't I add nitric acid? Why don't I add sulfuric acid? What are we going to do is we have to make sure that, for example, I have taken a piece of a liver or a piece of a or a leaf. So when I take a leaf or a liver, what happens is the cells are very well stitched together, correct, right? So they are attached together very well. So the stitching is so firm. So when I add trichloroacetic acid, what my trichloroacetic acid will do is it will try to dissolve all this stitching or the attachment or the intracellular cementing, what we call it as. So it will just dissolve it. So when it dissolves it, what happens? My cells will become free. From attachment, they will become what? They become free. So that will make me easy for it to turn it into a fine paste. So as I add two fractions of this trichloroacetic acid and I keep on grinding it, it becomes a very, very fine paste. So now what do I do is I transfer the fine paste that I've created after I grind it very, very well. So I collect this in a test tube. Before I collect this in a test tube, my fractions also contain some unwanted substances. So that is why I will filter it. I will use the filter paper, so one or a cheesecloth, and then I will filter the pool that I extract after grinding this very properly. So once I extract this, my test tube's gonna look like this. This is my test tube. I will be able to find this in two layers. If this is the whole of the liquid that I have collected, okay, here I can see two layers. One portion of it is soluble. What is soluble? Soluble is something that can dissolve, that is completely mixed up with the main fraction. What have we used? Trichloroacetic acid. So we have used acid in this. So some substances have completely dissolved in this acid, so they make this soluble or soluble pool, we call it as. Okay, we call it as soluble pool, like sugar and water. Like similarly, some substances have completely dissolved in this, so we call it as soluble pool. Whereas the rest of the substance, they float like oil on water. So what happens when we take oil on water? We can see that it's separated into two layers, right? We can easily differentiate them and we can also separate them. So this layer, which is not at all soluble in this acid pool, is called as acid insoluble pool, right? Insoluble pool. So we have done this all for a particular reason, and we are going to find out why did we even conduct this experimentation. So let's see what happens. Once I have the filtrate and it has separated into two different layers, one layer of a filtrate that is soluble and another layer of the filtrate is insoluble, what does it suggest me? If I do a chemical analysis of the soluble pool and insoluble pool, what I see is the acid soluble filtrate that I have, it contains something called as biomicromolecules. So in our nature, according to our human perspective, we can differentiate all the elements that are present, all the things that are present, into micro and macro based on the mass or composition or visibility, etc. Similarly, we are going to call this biomicromolecules for a particular reason. And the reason is these are the molecules that contain less than 1000 daltons of weight. If I measure any of these molecules, they are going to be measuring less than 1000 daltons of mass. Understood? So that is biomicromolecules. If I see the acid insoluble pool, that is called as retinate pool, if I look at them, what do I find? This acid insoluble fraction, they contain something called as biomacromolecules. So biomacromolecules means macro means more. That means their weight should be more than 1000 daltons. Simple as that. So when your questions come in your examination, see, a student took a tissue of a chicken, the liver tissue of a chicken, or a plant leaf material, and he adds trichloroacetic acid. After he grinds thoroughly and he separates and he takes and filters the entire filtrate, he is going to get two layers. One is acid soluble pool, other one is acid insoluble pool. Now the question is, what do these acid soluble and acid insoluble pool contain? Your answer: acid soluble pool contains biomicromolecules. And why? Because they are below 1000 daltons of weight, whereas acid insoluble pool contains biomacromolecules. Their weight is greater than 1000 daltons. Understood? So we have got these two pools, and we understood that there are some uniqueness in the composition of this pool. One pool says that it is soluble, one pool says that it is insoluble, and the solubility and the insolubility comes because of the composition. And we know that the composition of the soluble pool is biomicromolecules, and the composition of the insoluble pool is biomacromolecules, right? So did you guys understand this till now? Are you able to follow? Yes. Okay. All right. Thank you, students. Thank you for your response. Okay. So now that we have studied that acid soluble pool and acid insoluble pool contains biomicromolecules and biomacromolecules, right? So I appreciate that. So we have learned a little bit about it. So now what is really happening here? What is life and what is the capability of living organisms? And where do we get all these elements and compounds to construct things is a question mark. Let us say that I'm a vegetarian and I eat plants. When I eat plants, I'm going to gain some inorganic substances as well as organic substances. Organic substances in the form of, if I eat potato, I'm going to get some carbohydrates, which is again a sugar, correct, right? So if I eat the spinach, I'm going to get calcium. I'm going to get phosphates. I'm going to get potassium. So through food, I'm getting all these things. How did the plant get? The plant gets these minerals through soil. We know that very well. And the soil contains a lot of these minerals, okay, which are present. And also, they also get it through something called as dead and decaying matter, all these materials. So what is happening is, we are Earth itself. We are not special. Whatever that is the construct of our human body or any other living organism here on our planet, all of us are just like our Earth. So it's a constant cycle of recycling things. It's a constant cycle of movement of materials. What is happening? When we are alive, we eat plants, we use the natural resources around us, and we gather this cellular mass and we grow and we become strong. We age, we reproduce, and then we die, right? So as we are dead, what happens to our tissues? Do they disappear? No. They again go for the degradation cycle where we become the minerals again, what we have gathered. So what happens to this mineral? So the minerals are again taken up by these plants and the cycle continues. Correct, right? So this endless cycle where like we eat plants and plants eat us is never ending. What is the proof? The proof is, if I compare the human body to planet Earth, that is where the astonishing things will become evident to us. If I take a handful of soil, or the earth's crust and then compare it to the human body, look at the similarities that we have. We have hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, and silicon. All of this is inorganic material. And the earth crust contains, look here, they also contain hydrogen in percentage. Human body also contains hydrogen. Similarly, carbon, you compare to the weight to the earth to the weight to the body. Again, oxygen, nitrogen, and sulfur, sodium, calcium, magnesium, and silicon. All these elements, we similarly have the same elements but in different concentrations. Are we earth? Are we same as earth? Yes, you can say that I'm same as earth. Only thing is you are moving yourself, you are able to have the capacity of self-conscious, you have the capability of reproduction, you have the capability of movement, you have the capability of communication, etc. And earth gives you the capacity to possess all that by giving the same elements that circulate in your body and construct what you are. All right, so how do we know? Okay, so give me a second. So how do we know that all that we have, one second, yeah, all that we have from the earth is the same, right? So for that we have to go into a destructive process. So what are we going to do? Again, we are going to take a living tissue. So this is a wet tissue. So wet tissue means like when you extract it from, for example, if you just take a liver, it's going to be wet, isn't it? So now what we are going to do, we have to do it, we have to make it dry. Either you go for solar drying or heat, you dry it in the heat, or you use the oven to dry all these water molecules that are present in this tissue. Once we vaporize this water, what we are going to do is we are going to burn this. So when we burn this, it's going to release a carbon dioxide, water vapor, and it forms ash, white color ash is formed, right? So, excuse me. Yeah, white color ash is produced. If I take this ash and if I analyze what is present in this tissue, I am going to get the same compounds that we discussed. All these compounds, which are those? Which are hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, and silicon will be found in our living tissues. So that shows that we are same as earth. Look here, we are going to get elements like calcium, magnesium, sodium, potassium when we burn it. And inorganic compounds can also be found. These are elements and their compounds can also be found. Something like sulfate, phosphate, sodium chloride, calcium carbonate, etc. So we are very, very similar to that of what is in our surrounding. Correct, right? So we surroundings makes us. So that means we are surrounding itself. So that is why I told you this is a beautiful chapter to talk about, because this talks about the very core of life, what you are made up of. Okay. So let's go and study what is biomicromolecule. As I told you earlier, whichever the molecules that are having the weight less than 1000 daltons. So what should be the biomicromolecule? It should be less than, can you see the ink? Students, can you see the ink? Let me know. Is the ink visible? Yes. Fine, fine. All right. So let's go back. So if I see the biomicromolecules, as I told you, it should be less than 1000 daltons. Okay. So it should be less than 1000 dalton. So what is the actual weight that we are looking for? So usually our micromolecules, they range from 18 to 800 dalton. So their weight is almost around 18 to 800 dalton. So where are they present? Where did we find this? So usually to specify this where can we find biomicromolecules? We just go back to our experiment and just pinpoint that biomicromolecules were found in the acid soluble pool. That is where the first evidence that we have, okay, we have the differentiation, right? So they're found in acid soluble pool. So what does it represent? Where can they be found in the cells? So they are usually present in the cytoplasm, right? So these are all very, very, very important for your examination, students. So it's very essential that you note it down. So where are they present? Biomicromolecules make up the cytoplasmic composition of your cell. So should I explain what is a cytoplasm? Probably not. But still, if you see the cell within the plasma membrane, there is a uniform matrix that is present, right, within your cell, that holds all your cell organelles, that gives a proper, what do you tell, an even environment, or a very uniform environment for all the movement of these biomolecules and biochemical reactions, etc. So that is nothing but cytoplasm. And our cytoplasm is made up of biomicromolecules, which are having around 18 to 800 Dalton. We have spoken so many times about biomicromolecules, biomacromolecules. So now, what are these biomicromolecules? What are they made up of? So usually, if you just look at what are different types of biomicromolecules, you come across something called as amino acids, lipids, sugars, and nitrogen bases. So these are the four types of biomicromolecules that are found in all living organisms. So now we are going to discuss individually what are these and what are their structure, composition, and properties. Okay, little bit of chemistry and more of biology. All right, so let's go. So when we talk about the first biomicromolecule, that is amino acids, so amino acids, or amino acids, whatever you call, first of all, let us take a very, very basic step to understanding this. If I look at a living organism, let's say you're looking at a human body right now. So what are you made up of? You can tell cells, you can tell tissues, you can tell organs, organelles, whatever it is, right? So if you see your whole body's composition, at least 95 to sorry at least 95 to 98% of your body's biological functions is made up of proteins. Proteins are very, very important for everything, like to give you immunity, for your ability to fight against diseases, proteins, to construct your skin and muscles, proteins, to construct your bones, proteins, to signal your cells something, proteins. You understand my point? So for everything, 98% of the biological functions is done by proteins. What does your blood plasma made up of? Proteins. Again, proteins. What is your blood made up of? Blood cells. Red blood cells made up of hemoglobin. What is hemoglobin? Protein. What is cellulose? Cellulose is a sugar, sorry. So if you look at your most of the biological functions that in your body, it is made up of something called as proteins. You might have heard about your teacher or your friends or your family members, your mother and father scolding you, eat this food because you need protein. Why do you need protein? Answer is here. 98% of your biological functions are made up of protein. Okay, let me explain you very simply how to understand a protein. See, are you all aware of red blood cells? Okay, two questions. What are, what is red blood cells? What is the function of red blood cells? Let me see your answer. What is the function of red blood cells? Come on, answer, students. What is the function of red blood cells? What is the function of red blood cells? Do you have an answer, students? Come on. It's so simple question. Okay. Maybe you're taking time. Let me tell you. Do you know the function of red blood cells? You know the reactions? Yes, no. Come on. Give me the reaction. No? You don't know? Yeah, it carries oxygen, I think. Oh, yeah. Let's see. It carries oxygen and delivers to our body or tissues. Excellent. So, do you agree that to do this function, that to carry oxygen and deliver it to different parts of a body, we need hemoglobin? Agree? We need hemoglobin. Hemoglobin is within the red blood cells. Correct, right? So we used to study in our lower classes that red blood cells is made up of red color pigment containing hemoglobin. That is why it is red in color. Yes, correct. Have you heard about hemoglobin, students? Yes. See, we have an answer. So that's right. All of you are telling you. Very nice. So this is what I need. So all of you know that we have hemoglobin, and hemoglobin carries blood, I mean like oxygen, right? So what is hemoglobin? Hemoglobin is a protein, students. So we know that our blood cells contain iron, right? So we call it as ferrous protein or ferrous ion. So this, for this iron, we have this protein chains. So let us say that I will write a structure, not an actual structure of hemoglobin, but a depiction of hemoglobin molecule. Okay. So let's say that for this iron, there are 4 chains. Okay. There are 4 different chains. Okay. So what are these chains? If I look at a hemoglobin molecule, it will look like this: one iron protein in the one iron atom in the center or element in the center. To that, 4 chains are attached. What are these chains? I will name them as alpha alpha and then beta beta. Okay, alpha chain, beta chain. All right. So what are these alpha chain and alpha chain? Every protein that we talk about has a specific structure. Understood? So you don't have to worry about it. Just take like this, that this protein is a globulin protein because it has a globular structure like a globe. Okay. So that is why it is having 4 chains. And what are these 4 chains? What are these chains made up of is very important to us. So we can call this, we usually call this as globulin chain, alpha globulin chain, beta globulin chain, similar to that. Understood? So why did we discuss about this? Let us talk about this alpha globulin chain. If I just take alpha globulin chain and then I talk about it, what is it made up of? A chain is made up of monomers. Agree? It's a polymer. It is a polymer. It is made up of several monomers attached together. That is what has made it a chain. So what is its monomer? So if I attach what is called as an amino acid, okay, one after another, I will get a series of amino acid which is in the form of a chain. I call this as a peptide chain. I call this as a peptide chain. So each peptide chain is made up of a sequence of monomers of amino acids that are sequentially arranged. Understood? So we understood our protein is made up of chains, and we can call these chains as peptide chains, and our peptide chains are made up of monomers of what? Amino acids. Understood? So when you arrange amino acids in a specific sequence, we get a peptide chain. When we take different peptide chains and combine, we get a protein. Simple. Understood? Yeah. So let's understand now what is an amino acid. Now, to understand an amino acid is so easy, students. Okay, absolutely easy, and I'll guarantee you this. Okay, so how do we understand an amino acid? Step one, we have to write a methane. So what's the formula for methane? CH4. So let us take a carbon atom, and I will give this carbon atom 4 hydrogen atoms. Understood? So I'll give them 4 hydrogen atoms. We have 4 hydrogen atoms. Now what we have to do is we have to knock them out. We have to knock them out. How do we knock them out? Okay, I'll just take out this. Sorry, I'll just remove this hydrogen atom. Done, gone. And I will give this. I will replace this hydrogen atom with COO minus. Okay, I will remove this hydrogen atom. Sorry, I will remove this hydrogen atom, and I will replace it with NH2. Okay, and I will remove this I will remove this hydrogen atom, and I will replace it with an R group. What did I just do? I created an amino acid. Simple. They say that amino acid was created by the substitution of hydrogen atoms that was initially present in a methane. So methane was CH4. So what happened with this CH4 was the hydrogen atom got replaced by certain different types of elements or compounds. So what are those compounds? One of the hydrogen atom got knocked out and an amino group started adding up. This is called as amino group. Understood? and another hydrogen atom got knocked out, and here a carboxyl group added. Another hydrogen atom got knocked out, and a functional group replaced. So this functional group can be anything. We'll discuss about it. What can be the functional group and what's the possibility? We'll discuss it. Anything that gives a meaning and a particular characteristic for an amino acid is called as a functional group. And another hydrogen atom said, I'm not going anywhere, I'm going to stay here. So this is the general structure of an amino acid. So I know that it is very messy on the board. So let me get rid of this. Okay. Sorry. Excuse me, students. Yeah. So I'll just get rid of this. So if I get rid of this, to your right side of your screen, you can see a beautiful structure of our amino acid. What do we have here? We have a central carbon atom, an alpha carbon atom. To an alpha carbon atom, we have an NH2 group. This is called as N-terminus or N-terminal or N-end. We can call it as N-end or N-terminal. So if you see here, COOH or COO minus, so this is called as carboxyl group. This is called as a functional group, and this is a hydrogen atom that is present. So this is a general structure of an amino acid. So whenever two amino acids interact with each other, what happens is an N-terminal of an amino acid of one, see, we have one amino acid here, and we have another amino acid here. When I say N-terminal of one amino acid, that means this end. When I say C-terminal of an amino acid, that means this end. So the N-terminal of one amino acid combines with the C-terminal of another amino acid through a peptide linkage. A peptide bond is made between this N and C. Got it? So then what do we have? We have one amino acid connecting with another amino acid through a peptide linkage. So now they're together. So we have what? We have a chain of amino acids following a peptide chain. Did you understand? Yes? Did you understand the general structure of amino acid? One. Yes, perfect. Others, you didn't understand? Yeah, good. So the pace of the class is fine. You're able to follow it up. Yeah, good. Excellent. All right. Nice, students. Okay, you're replying, and it's very happy. I know I keep repeating this because it's really the story of it. Okay, chalo. So let's go for the next topic. We are going to discuss about amino acids only, but we are not going to leave it by understanding the general structure. General structure only gives us an idea about what is the composition of this molecule. If I talk about amino acid, what is amino acid made up of? So if a question is asked, now you are very smart, you're going to answer. See, amino acid is nothing but a central carbon atom connected to an amino group, a carboxyl group, a hydrogen group, and a functional group. This is what will be your answer. Do you agree with me? Yes. So before this, just few minutes back, if I asked you what is amino acid, some will tell, I don't know. Some will tell a monomer of a polypeptide chain or a monomer of a protein, you will tell. But would you tell this general structure? No. Maybe yes. If you tell yes, genius. If you didn't know, good. Now you know. All right. So let's go next. And I told you the R group of an amino acid can be anything, right? But it has to give meaning and a property to that particular amino acid. So let's say that our R group is a H or a hydrogen atom. So a hydrogen atom, say for example, here is a normal hydrogen atom that was present. Here is an N group. Here is a C group. And here you see a hydrogen atom has taken place in the place of R. R is defined by an hydrogen atom. So the functional group of this particular amino acid is H. So example is glycine. Glycine is a name of an amino acid. Similarly, we have R group has CH3. So you can see here CH3 is the R group. So here what do we have here? We have alanine is an example. Next one is R group is CH2OH. So the example is serine. So you can see here a OH group is here, and the structure is quite complicated, but it's fine. We have a CH2OH. Sorry, CH2OH. All right. So these are the functional group. So if a H group is functional, then the example is glycine. If a R group is CH3, then it is alanine. If a R group is CH2OH, then it is serine. So these are the examples of amino acids based on their functional group. Now, if you look at the overall number of amino acids in our nature, in our living organisms, we come across 20 different types of amino acids. There are many types of amino acids apart from this 20 type, but in living organisms, especially for synthesis of proteins, I told you, right? Protein synthesis is major. So for this only we are studying amino acids. So for synthesis of protein in all living organisms, the living organisms use around 20 different types of amino acids. So among these 20 different types of amino acids, based on these characteristics, chemical nature, we can divide them into 3 types. What are those? We have acidic amino acids, we have basic amino acids, we have neutral amino acids. Simple. Any chemical compound can be divided into 3 different groups: acidic groups, basic groups, and then you have neutral groups. Correct, right? So if you look at acidic group, the example, you have to take down these examples and keep on revising it. Enjoy the process. Okay, don't take things like, oh, I have to remember so much. No, just when you just give your heart to amino acids give its heart to you. Okay, acidic amino acids example is glutamic acid. Basic amino acid example is lysine. Neutral amino acid example is valine. And there are other groups of amino acids also, like such as aromatic amino acids, which gives you a nice smell. Okay, aromatic amino acids. For example, tyrosine, phenylalanine, and then we have tryptophan. I'll write it here. We have something called as tyrosine. Okay. Next one is phenylalanine. And then is tryptophan. Okay. So these 3 come under aromatic amino acids. Aromatic amino acids means smell. Okay, get a smell. Why are we discussing this slide? We are discussing because we need to understand, do amino acids give a particular chemical characteristics? Do they have a chemical characteristic? For example, if I touch water, then it is neutral. If I take sodium hydroxide, it is basic. If I take HCl, it is acidic. Similarly, when you talk about chemical compounds, they have to exhibit some chemistry. So the chemistry of amino acids also being exhibited through acidic, basic, and neutral characteristics. That means there are different types of amino acids based on its chemical nature too. So the examples are glutamic acid for acidic, lysine for basic, neutral for valine. And we also come across aromatic compounds such as phenylalanine, tyrosine, and tryptophan. That is why we had to discuss about this. All right. So, students, chalo, we'll talk about essential and non-essential amino acid in the next class. All right. So we will end our session. Okay. All right. So I will see you tomorrow at the same time, and we are going to continue our journey studying about amino acids. Okay. Thank you very much for attending the first session. It is so encouraging to see you in the class. Perfect. Okay. I'll see you tomorrow, same time. Don't miss the class. Be regular. Okay. Bye-bye. Take care.